In this video, we're going to look at DNA recombination. So we're just going to start over with some general terminology about crossing over. And so when we talk about combination, you could be talking about homologous recombination or general recombination, where DNA segments with extensive homology will cross over and exchange DNA. You can also have site-specific recombination, where two short specific DNA sequences will cross over. So both of these talking about you need homology generally for crossing over to happen. Now, when you're doing crossing over, you can exchange genes between homologous chromosomes, and that's what's being shown here. So A is the actual picture of that happening, B is the colorized version of that. And so we have two chromosomes, orange and blue, and you can see that we are exchanging DNA, or crossover events happening right here. Now, this can only happen, this representation, in organisms that have multiple chromosomes. So bacteria, they just have one copy of circular DNA, so they really can't do this, but they have their own mechanism for recombining genes. Now, it is also possible if we have a foreign gene in otherwise homologous DNA, that is a way that this foreign DNA can be installed into the genome as well. Now, to think about recombination, we're going to look at the model that comes from Holliday, and Holliday is the name of the person who made this model. It doesn't talk about like holidays in general. So this is what we're looking at, where we have two chromosomes, blue and red, and we're also going to designate them capital letters and lowercase letters. So first, we line up our homologous chromosomes. Then a nick is made into each chromosome. After that nick is made, the strands will cross over, and you'll have a four-stranded structure known as the Holliday junction. So this Holliday junction, once it's made, it can move along the chromosome. This is called branch migration. So as long as they're connected, this Holliday junction can just move left to right in either direction. Now, once we made our Holliday junction, we have to eventually cut it again, because we don't want our chromosomes, our blue and red chromosomes, to be linked forever. So what we first need to do is just rotate our DNA to resolve our X, and now we need to think of it like a four-way intersection. So it's much easier to think of resolving this once we think of it in this idea. All right, so there's two ways to resolve this. One, we cut the strand that crossed over, or we cut the strands that didn't cross over, so that's left and right. All right, so let's handle the left case first. We cut the strands that crossed over, and the way that you're going to seal this is that if we made a cut for the strands that crossed over, if you follow my cursor, we're going to reseal it by going on the red and taking a left turn. If I'm moving down this way, I'm taking a left turn. So in your mind, you're picturing my cursor, and then I'm going to seal it to this strand. In the opposite direction, seal it to that strand. So that's what's going on. And you can see when we do that, we'll have one chromosome that's all blue, and our other chromosome here, we have switched for the red gene. And we did that same switch in the chromosome that's lowercase. Well, let's imagine what happened if we cut the strands that did not cross over. So here, follow my cursor, I'm going to connect to this strand. And the same thing here. For the lowercase, I'm going to connect to this big case B. So that's how we're going to reconnect the DNA. And you can see when we do this, we have actually exchanged DNA on both chromosomes. So blue, now I'm going to continue with the red. And same thing on my other chromosome. So one part is exchanging one gene while the other cut is exchanging like the whole other half of the chromosome with each other. But that is two different ways we can resolve this holiday junction. And that's it for this video. I will see you in the next video.